Hello, everyone. My name is Anna Zerio. I completed my PhD at Balliol College and in cardiovascular physiology at the Department of Physiology, Anatomy and Genetics. And it's a great pleasure for me to introduce you to this lecture today by Professor Manuela Zaccolo. Manuela is not only an exceptional scientist, but I was lucky enough to have her as my supervisor. So it's a particular pleasure to be moderating this lecture today. A few words about Manuela. Manuela Zaccolo is a cell physiologist distinguished internationally for her groundbreaking work on intracellular signaling. She has made major discoveries leading to our current understanding on how second messenger achieve hormonal specificity via subcellular compartmentalization and nanodomain signaling. This novel paradigm has profoundly changed the way we study intracellular signaling and the way we think about the involvement of second messenger in disease. It has provided a new perspective for the development of approaches that target signaling pathway in therapeutic purposes. Manuela has authored 197 publications and her work has been cited over 10,000 times. Manuela graduated in medicine at the University of Torino in Italy and subsequently went on to pursue a career in science by spending 4 years as a postdoctoral researcher at the Laboratory of Molecular Biology in Cambridge, UK, working on protein engineering and in vitro molecular evolution. She then moved back to Italy at the University of Padova to work on the generation of fluorescent sensors for real-time imaging of intracellular second messengers in living cells. In Padova, she established her independent research group in 2001 at the Venetian Institute of Molecular Medicine with a focus of intracellular signaling. In 2007, she moved to the University of Glasgow, where she initially held a position as a reader and subsequently as a professor of cell biology. And in 2012, she joined the Department of Physiology, Anatomy and Genetics at the University of Oxford, where she's a deputy head of department and director of the Burden-Sanderson Cardiac Science Centre. Manuela is an elected member of the Accademia Europea, elected fellow of the Royal Society of Biology, and elected fellow of the Istituto Veneto di Scienza, Lettere e Arti. In Balliol, she is a fellow and a tutor in preclinical medicine. And I will hand it over to Manuela and her lecture, which I hope you will enjoy as much as I did with my project.
Thank you very much, Anna. I will just share my screen. So yeah, thank you very much, Anna, for the introduction, and hello, everyone, and thank you for joining this online lecture today. What I would like to tell you about today is a project that started over 20 years ago now, and that's with my research group. We are now finally bringing closer to practical applications. I'd like to say that I kept my presentation fairly light on the technical details, but I'd be very happy to dive deeper during the Q&A at the end if anyone is interested. So in my laboratory, we look at how the heart reacts to stress, the so-called fight or flight response that you probably heard of. And we try to use these insights, what we learn by studying this signaling system to come up with better ways to treat cardiac disease. And so I will take you through, during my presentation, I will take you through some of the key steps that have brought us to a point now where a clear path toward clinical translation is finally in view. So what is the fight or flight response? As you probably know, this is a physiological response that is triggered when we sense the presence of danger or we are in a situation of physical or emotional stress. And in this situation, our brain triggers the release of a hormone, adrenaline, And adrenaline distributes across the body and acts on many different organs and systems to prepare the body to react to this situation of danger. But at the level of the heart, what adrenaline does is to make the heart beat faster and more forcefully, increasing in this way the flow, the blood flow, to prepare the body to react to the situation of danger. This signaling system, however, is not only active, doesn't kick in just when we are in an emergency situation. It is also active, for example, when we exercise, but also during simpler things like climbing the stairs or speeding up to catch the bus, or simply when we get up from a chair. So in all those situations where the heart has to work just a little harder. And this is a common knowledge, and all of you will have experienced either directly or because people that suffer from heart disease, that when someone has a heart condition, these simple activities suddenly become very tiring and very hard to do. And this is because the heart, when there is a pathology, doesn't respond to adrenaline as smoothly as when the heart is healthy. So how does the fight or flight response work in the heart? When the brain triggers this signal to release the stress hormone adrenaline, the adrenaline distributes across the body and reaches the surface of the cells that make up the majority of the heart mass. We call these cardiac myocytes. These are muscle cells, and they are responsible for the contraction and the relaxation of the heart. So when the adrenaline comes, if we focus here and we zoom in on these regions, this small region of the cell, the adrenaline finds a receptor, so a protein on the membrane, on the external membrane of these cells, and adrenaline binds to this receptor. And this interaction triggers the synthesis of a chemical. This is called cyclic adenosine monophosphate, or cyclic AMP in brief. And this chemical, this small molecule, works as an intracellular messenger, and it goes on to activate an enzyme called pKa. And pKa's job is to add a small chemical tag, a phosphate group, to other proteins. And by doing this, this changes the way these other proteins behave. And these are the proteins that control how the heart contracts and how the heart relaxes. One important other element here is this group of enzymes. These are called phosphodiesterases. And these are the enzymes that can destroy cyclic AMP. They hydrolyze cyclic AMP, so they get rid of this second messenger. And so these phosphodiesterases are responsible for switching off the signal. So how does this signaling system work in the heart? And to explain this, I need to take you for a quick trip inside the cardiac myocyte. So the heart is made of millions of these tiny little muscle cells. And these are the cells that actually do the squeezing and the pumping. Inside each of these cells, if we zoom in here, we can see there are neatly organized bundles of proteins arranged in kind of long cables that we call myofibrils. And if we zoom in even further here, we can see that each myofibril is built from repeating sections linked end to end. And each of these sections is made of two strands of proteins, the thin filament and the thick filament. And these two strands of proteins slide over each other And this sliding motion is what is the basic mechanism that allows the heart to contract and to relax. So the heart pumps out the blood during contraction. And after contraction, the heart relaxes. And this is equally important because during relaxation, the heart can fill with blood so that there is blood in its chamber ready for the next contraction and for the next ejection of blood. So every heartbeat involves a contraction and a relaxation, and this repeats over and over about 3 billion times over an average lifetime. And as I mentioned in the previous slide, contraction and relaxation is due to the sliding over each other of these thin and thick filaments. And you can picture, to understand the mechanism, you can picture two teams facing each other, each holding the same rope. They grab the rope and pull. However, they do not pull the rope towards themselves. Instead, what they do is that they pull themselves closer along the rope. As both sides do the same, so this the distance between these two teams shrinks, and this is what happens during contraction. Then they let the rope go, and the sliding mechanism leads to the relaxation of these elements in the resting condition, as shown here. Now, one important thing that you need to know is that the grabbing of the rope requires calcium. We are focusing here on these two elements, one thin filament and one thick filament. And for the thin filaments here, there contains these other elements. This is the multiprotein complex called the troponin complex. And this troponin complex controls how calcium binds to the sliding filaments. And as I said, calcium binding is absolutely required to trigger contraction. If calcium is not bound to the thin filament, the thick filament cannot grab. And during relaxation, this calcium needs to be released. And it's only the release of this calcium that will allow the filament to slide back to the resting state and therefore to relax the heart. And here I would like you to make note of these elements shown in purple. So this is one of the components of the troponin complex. This is called troponin I. And I want to notice this particular component because this will be a very important actor in my story. Okay, so let's go back to the fight or flight response. When the brain triggers the release of adrenaline, as I said, adrenaline travels across the body, reaches the membrane of the cardiac myocytes, and there it binds to its specific receptor. This is called a beta receptor. And you may know these receptors because of drugs that are called beta blockers. And these are drugs that are very commonly prescribed for the treatment of chronic high pressure or for the treatment of certain heart conditions and other pathologies. So adrenaline binds to the receptor, and this triggers the synthesis of this chemical cyclic AMP. Cyclic AMP goes on to activate pKa, and pKa phosphorylates, adds this phosphate group to a number of proteins that are critical for the control of contraction and relaxation of these cells. As I mentioned before, one of these targets is troponin I. So pKa can phosphorylate troponin I, and this phosphorylation reduces the affinity for of the myofilament for calcium. So this phosphorylation will promote relaxation. On the other hand, phosphorylation of these other targets has the effect of increasing the amount of calcium that is available during each contraction for binding to the myofilaments. So these other modifications, the modification of these other proteins, promote contraction. When we started working on this project many years ago now, we were concerned with one specific problem. We were trying to address one specific question. So we knew that protein kinase A is a very promiscuous enzyme. And in cardiac myocytes, in addition to phosphorylating these proteins that are involved in the regulation of contraction and relaxation, we knew that pKa could phosphorylate a very large number of other proteins. Some of these are receptors or channels. Some proteins are involved in the control of metabolism or in the control of cell growth or cell death. A large number of proteins that are localized to the nucleus where this phosphorylation modulates the transcription of genes. In addition, we knew at the time that the beta receptor is not the only receptor that signal via generation of cyclic AMP, but cardiac myocytes express on their surface a large number of other receptors that are activated by different hormones, different from adrenaline, and hormones that do not modulate the contraction and the relaxation of the heart. However, all these other receptors, they also signal via the generation of this same chemical, cyclic AMP. So the question we were trying to address at the time was a question that had been remained unanswered for many decades since the discovery of cyclic AMP, which is, how can this happen? How can different hormones result in different cellular effects when they all signal through the same molecule, cyclic AMP? And the first breakthrough came when we were able to develop a tool, a probe to measure cyclic AMP in intact living cells. So this probe is a fluorescent indicator that we can put into cells, any cell type. And this, in this example, this is a cardiac myocyte. So we can express this sensor or express, insert these sensors into living intact cells. And we can monitor in real time the changes in the second messenger cyclic AMP as it happens in the complexity of the intact cell. And so these images here are represented in pseudo color, and a dark color, blue, green, means low cyclic AMP, and the color turning yellow and red means an elevation of cyclic AMP. So here you see that in this experiment, we treated the cells with norepinephrine, which is similar to adrenaline. And these, as we expected, generated an elevation of cyclic AMP. But what we demonstrated with this experiment is that, contrary to what the field thought at the time, and the elevation of cyclic AMP does not happen homogeneously throughout the cell. You can see that better in this enlargement here. But the elevation of cyclic AMP happens in discrete compartments. So if we now focus on this schematic here, this means that when a receptor that triggers the synthesis of cyclic AMP generates an elevation that is higher in certain subcellular location, as higher than in other locations, and there will be some locations where the signal is not detected at all. And the important thing is that the pattern of these cyclic AMP gradients will depend on the specific hormone that has triggered the production of this signal, so that different hormones will generate different gradients in different locations of the cell. And in this way, different proteins will receive that phosphate group and will be able to then go on and modify different cellular functions. So basically, in this way, through compartmentalization of the signal, different hormones can mediate different cellular effects. The other important findings in these experiments was that we demonstrated that these gradients are shaped by the activity of phosphodiesterases. Remember, the phosphodiesterases are the enzymes that can degrade cyclic AMP. And so different phosphodiesterases degrade to a different extent cyclic AMP from different locations inside the cell, and this generates these gradients of cyclic AMP. And this is demonstrated here, where we inhibited the activity of these phosphodiesterases using this drug IBMX. And you can see that when the activity of this enzyme that hydrolyze cyclic AMP is blocked, now cyclic AMP can, the boundaries between these compartments are abolished, and cyclic AMP can diffuse more homogeneously in the cell. Okay, so to reiterate, this was the first key discovery that led to the current project. What the field initially thought was a homogeneous signal generated by the fight-or-flight response in the cardiac myocyte. When we looked in more details using our fluorescent probe, we realized that, in fact, this signal is made up of a multitude of highly compartmentalized tiny signals, each contained in a very small domain with a dimension of a few nanometers. And the nanometer, for those of you that don't have this clear, is a millionth of a millimeter. So here I'm only representing 3 of these domains, but there are many, many others spread throughout the cell. And each of these domains has a very specific localization within the cell, reaches a specific level of the message and a particular duration, a very specific duration of the message. And each of these domains is in charge of modulating one specific cellular effect. And we quickly became interested in one specific domain, or nanodomain, which is a domain that is centered here around the troponin complex. As I mentioned earlier, the troponin complex is important to modulate relaxation because elevation of cyclic AMP activates PKA. PKA puts this phosphate group on the troponin I component, and this reduces the affinity for calcium. So calcium can be released more easily, and this promotes relaxation. So we became interested particularly in this domain, and we did an experiment here where we wanted to compare the cyclic AMP signal at the troponin complex with the cyclic AMP signal generated at 2 other locations. One is here at this other membrane. This is called sarcoplasmic reticulum, and it's illustrated a bit better in this schematic here. This is a kind of reticular, it's made up of a reticulum of cisternae delimited by a membrane, and these cisternae are a reservoir for calcium. So these cisternae contain a very high concentration of calcium, and this calcium is released during each contraction, and this is the calcium that then binds to the myofilament. This protein here is also phosphorylated by protein kinase A, so it's under the regulation of cyclic AMP. And the third compartment we wanted to monitor is this other protein here called myosin binding protein C, and this kind of links the thick and the thin filament. It's also phosphorylated by PKA, and again is involved in promoting contraction. So we have one domain here involved in promotion of relaxation, 2 domains here involved in promotion of contraction. And when we did the experiment using these sensors and we stimulated the cell with adrenaline and measured the cyclic AMP levels at the 3 locations, this is what we found. So first of all, we confirmed that cyclic AMP, the cyclic AMP response is compartmentalized. The response is very different at the 3 locations, but we were particularly intrigued to see that the response at the troponin complex not only is smaller than at the other 2 locations, but we were particularly interested to see that it is very transient in nature. So this suggests that at the troponin complex, there is a specific regulation that turns the signal off quickly. And I draw your attention to the fact that the distance between these structures is only about 30 nanometers. And so the fact that this transient regulation happens only at the troponin complex and not at the myosin binding protein C site really indicates that this regulatory mechanism is extremely focused around this troponin complex. And so that there is a very precise and focused control of cardiac relaxation. We, of course, wanted to understand what is the mechanism that determines this very focused relaxation, the control of relaxation, and fast forward a few years, we discovered that this transient response at the troponin complex is due to the presence of a phosphodiesterase that directly interacts with the troponin complex. So at this location, this phosphodiesterase can degrade cyclic AMP, reduce the activation of pKa, and reduce the phosphorylation of troponin I. So that this kind of puts a brake on relaxation because it makes the release of calcium a little bit difficult and makes calcium to stick to the thin filament a little bit better. But the real astonishing surprise for us came when we looked at diseased hearts. When we looked at the heart of mice where we induced heart failure, we found that the amount of these signals of this enzyme that switches off the cyclic AMP signal at this location was much higher than in healthy hearts. Up to 5 times more PDE was found in failing hearts compared to healthy hearts. So in these animals with heart failure, then the mechanism that regulates relaxation becomes massively overactive. The degradation of cyclic AMP is excessive. There is very little phosphorylation of troponin I. And calcium remains bound to the myofilaments and resulting in a muscle that tightens. And this is a major problem because when the heart doesn't relax properly, it will not fill properly with blood before the next beat. And if it doesn't fill properly, then it will not pump out a sufficient amount of blood in the next contraction. So this got us thinking, what if we could displace this excessive amount of phosphodiesterases in the diseased heart? Because if we could do that, then we could boost cyclic AMP levels right where that's needed, so around the troponin complex. And this would lead to elevation of cyclic AMP, more activation of pKa, increased phosphorylation of this troponin I, and facilitated release of calcium from the myofilaments. And this would enhance relaxation. And that's exactly what we did. Again, fast forward a couple of years, and we developed a peptide that we called P33 that does just that. It displaces the excess phosphodiesterase from the troponin complex. And the beauty of this approach is in its absolute precision. So P33 very selectively only interferes with the interaction of the PDE with the troponin complex. And it only induces an elevation of cyclic AMP in this tiny nanodomain, a few tens of nanometers around this troponin complex. And the P33 has no impact on the level of cyclic AMP that we can detect at the protein on the sarcoplasmic reticulum, or the myosin-binding protein C on the thick filament, as shown in these graphs here. So here I'm showing the level of cyclic AMP measured at the troponin complex in the absence of the peptide, or with a peptide that acts as a negative control. We see the typical transient response. But here in dark blue is the cyclic AMP elevation in the presence of P33. You can see when we displace the PDE from this complex, we induce an elevated and sustained increase in cyclic AMP at this location. But P33 has no effect whatsoever on the level of cyclic AMP at the myosin-binding protein C or at the circa complex measured by this other sensor. So the next question was, obviously, could this work as a therapeutic approach to fix impaired relaxation in diseased hearts? And to test this idea, we went back to our mouse model of heart failure. So the model we are using here is a mouse where the aortic arch is partially restricted with a string, so that the heart needs to push to pump against that increased pressure. And this mimics very closely what happens in humans that develop heart failure as a consequence of chronic high blood pressure. So we developed the model. We induced heart failure in these mice, and then we injected these mice with either P33 or a control peptide. And I apologize, this slide is a bit complex, but here I'm showing sections of the heart, and these are healthy hearts, and this is a heart where we induced heart failure, and where the animal was injected with the control peptide, where we expect no beneficial effect. And the blue staining here that you can see in this panel is the staining for fibrotic tissue, which is very abundant in failing hearts because they tend to remodel and to generate scar tissue. And here is a section of a failing heart from an animal that we treated with P33. So you can see there is a very significant reduction in this blue color, which indicates a very significant reduction in the fibrotic tissue. And that's quantified here. There is about a 50% reduction in the amount of fibrosis when we treat the diseased animal with P33. We also could demonstrate that in the animals treated with P33, here there is a complete recovery of this parameter, which is called left ventricular end diastolic pressure. And this is a parameter that directly measures the ability of the heart to relax. So in the failing heart, the mice with heart failure treated with the control peptide, this is elevated compared to the healthy animals. But the diseased animal treated with P33, we have a complete recovery of this parameter. And also we could show that the diseased animal treated with P33 also recovered their ability to pump blood outside the heart. So this is a measure of the ejection fraction. And this is a consequence of the fact that the heart can relax better and so can fill with more blood, and so more blood can be ejected at the next beat. The next question, of course, was, does this matter at all for humans? And the answer is yes, it does. So here we are looking at cardiac tissue that we obtained either from healthy donors or from patients affected by heart failure. And here we are measuring the amount of PDE that is in complex with the troponin. And you can see, as we found in the animal models, there is at least around twice as much PDE in complex with the troponin in fading hearts compared to LP. And this correlates with a reduction, 50% reduction in the level of phosphorylation of the troponin in the fading hearts compared to the LP. And with human cardiac myocytes, this is an experiment that we did in vitro, of course. With human cells, we cannot test at this stage our strategy in vivo. Of course, so we isolated cardiac myocytes from these samples that we received from cardiac surgery, samples obtained from heart failure patients. And we treated these myocytes in a dish with P33 to displace the PDE from the troponin complex. And we can show here that when we measure the local level of cyclic AMP, again, when we use the control peptides, that is a negative control, we see this typical transient response. But when we treat the cells with the P33, we see a larger elevation and the sustained elevation of cyclic AMP specifically at this location. And so the same mechanism that works, that we described for the animal models also works in human cardiac myocytes. So this is super exciting for us because heart failure is a major clinical problem. There is an estimated 60 million people affected by this condition worldwide. And heart failure is still the first cause of hospitalization and death. And 1 in 5 people may expect to develop heart failure during their lifetime. So this is a major burden on the health system and on society in general. So now when we think about heart failure, we typically think about a weak heart, kind of an exhausted pump that cannot push blood forward. And for decades, we have developed drugs almost exclusively to help with the contraction. However, there are cases of heart failure where the problem is not the pushing, is not the contraction, but on the contrary, the problem is the relaxation. And this type of heart failure is called heart failure with preserved ejection fraction, also called HFpEF. Where, as I said, the problem is not the contraction at all. The problem is the relaxation. And this is not a rare or minor issue because these make up about 50% of all cases of heart failure. And this type of heart failure is particularly common in people that suffer from diabetes or from high blood pressure, particularly common in women, and is very common in the aging population, which indicates that with the extension of the life expectancy, we may expect this type of heart failure to become more and more prevalent in the coming years. And one important point here to stress is that while the current drugs, the current medications that are effective in those forms of heart failure where the problem is the contraction, and that are quite good. So we can treat people where heart failure is a problem of contraction quite well, but those drugs do not work in those patients where the problem is the relaxation rather than the contraction. And so we believe that with our P33 peptide, or most likely an improved version of this peptide, we could potentially provide a strategy to fix this major form of cardiac disease that currently has no effective treatment. And of course, we are working towards this, although the path to translate our discoveries into potential clinical applications is clearly still very long. So with my last slide here, I'd like to make a final point and to say that the strategy that I described that that is the targeting of specific protein-protein interactions to modulate locally the level of cyclic AMP in a tiny, tiny volume inside the cell. We believe really this may represent a fundamental shift in how we think about drug design. Cyclic AMP is a very important signaling molecule, not only in cardiac myocytes, but in any other cell across our body. And alteration in signaling mediated by this chemical are known to underpin many other pathological conditions in addition to heart failure or cardiac disease. For example, neurodegenerative or cognitive disorders, diabetes, cancer, to mention just a few. So our idea is that rather than targeting the receptor that triggers the synthesis of cyclic AMP, and I should say at this point that a very large proportion of all the drugs that are currently on the market are drugs that modulate signaling mediated by cyclic AMP, but they do so by hitting the receptor, either activate the receptors or block these receptors. But when the drug modulates the activity of the receptor, this will affect all the domains downstream. And so this may lead to the effect that we are looking for, but it will definitely also affect other domains that we may not want to touch at all, because in fact, touching these other domains may be deleterious. And so we think that with our approach, where we very specifically target molecular interaction at specific subcellular locations, this should be able to deliver drugs with much higher specificity and which drastically reduce side effects. And the other point I want to make is that our discovery also suggests that many diseases may not be caused by proteins that do not work properly, but by proteins that are in the wrong place in the cell or are interacting with wrong partners. And this also opens a new way of thinking about disease mechanisms. And the way I like to think about this is that we may be entering an era where we aim at developing drugs that target specific molecular mechanisms with nanometer precision. And this, and to conclude, I'd like to acknowledge the members of my group, present and past members. This is us on a day out at Blenheim Palace, I think perhaps a couple of years ago. These are the people that carried out all the hard work behind the discoveries that I just described. And I really have been privileged to work over the years with an extraordinary group of individuals, and I'm deeply grateful for their intellectual contribution and their hard work and dedication. So thank you.
Thank you so much, Manuela. That was a great talk. And whilst we wait for a few questions to pop up in the Q&A box, and please feel free to send them over. I have first of all, a question. You mentioned drugs and you mentioned peptides. What's the difference between the two?
So drug is, by drug, we typically mean a small molecule. A peptide is what we in the field call the biologic. Is a bigger structure, is a bigger molecule, and has some potentially some more problematic applications in that a peptide can perhaps more easily be degraded once introduced in the body, and compared to a small molecule. And also it may have more difficulties in crossing the membranes to reach those intracellular targets. In more recent years, the research around optimization of peptides to be used as therapeutics has massively expanded, and now there is a very strong interest even from big pharma to develop this type of peptide-based therapeutics.
Thank you. And the second question that I had is that you were talking about treating the mice with a peptide. At what point, if we imagine that we translate this into the clinic, at what point will the patient receive this treatment? Is it when it's having an episode of heart failure, or is it preventative, or is it before? So what is the timescale?
Well, in the animal models, of course, we can decide when to inject the peptide, and typically one starts with injecting the peptide very early after the microsurgery, so before the full-fledged disease has developed, to see whether we can prevent the disease, because that gives us the better chance to see an effect. Of course, in patients, if the target patient population here is individuals affected by heart failure, the treatment would be delivered when the symptoms appear. So the peptide needs to be good enough not only to prevent the condition, but to reverse, or at least stop, or even better, reverse the condition. And all of this work is work that we need to carry out to, as we progress in this very long and very expensive and very time-consuming path towards translation.
Thank you very much. Just to some of the questions in the chat, John asks, how was P33 developed?
So the way we developed P33 was by looking at how the PDE and the troponin interact with each other. And then we designed the peptide. So we established which part of this molecule interact with each other. And then we designed a peptide that kind of replicate the amino acid sequence in the troponin. And we deliver this peptide in excess. So this peptide will work as a decoy. It will latch onto the PDE, and this will compete for the interaction of the PDE with the troponin, and will determine the inability of the PDE to interact with the full-length troponin.
Thank you very much. Alex asks, fascinating talk, thank you. Do we know anything about what occurs in these signaling pathways during cardiac arrest?
So this is a condition that we have not looked at, but cardiac arrest can be caused by an excessive release of adrenaline. And this is quite common. Well, it's not quite common, but it's a well-known condition. So you have a supercharge of the stimulation of all these components that are involved in the regulation of excitation contraction coupling. And one other aspect of the regulation of cardiac function through catecholamines, so adrenaline and noradrenaline, is at the level of the regulation of the rate of contraction. And again, there a disequilibrium of the cyclic AMP-dependent regulation can lead to severe problems with the rhythm and potentially leading to cardiac arrest.
Thank you very much. Could it be, sorry to jump in, but would it be possible that by altering the interactions with PD33, then you change a response in a different pathology? If every signalosome has an effect and is responsible for a specific function in, for example, heart failure, could you be modulating by changing that another downstream pathway in a different pathology or physiology?
You mean in a different cardiac pathology or in a different...
Yes, for example.
Yeah. So yeah, that's possible. So that at that point then would be a matter of dosing the amount of peptide that one want to deliver to the patients in a way that we don't overdo it. I think because this interaction between the PDE and the troponin is absolutely specific. So it's very unlikely, we think, that the peptide will have some off-target effect. Although that, you cannot completely exclude that until you do the experiments. But I think the chances that this peptide P33 will non-specifically affect other protein-protein interactions is quite small.
And that is the reason why you use a peptide rather than just using a drug that would otherwise have
Yeah, exactly. Small molecule will have much bigger chance of having off-target effect.
Anthony asks, will we be able to obtain a copy of the lecture, which was most interesting? Yes, absolutely. It will be onto the YouTube channel from Balliol College, Oxford, under Balliol Online Lectures. Emma asks, thank you for your talk. The mouse model had heart failure due to an aortic band. This would have produced HREF, but you are showing improved relaxation, which would improve HPEF. Does this suggest that the treatment for HREF, this is the treatment for HREF as well as for HPEF?
I think so. And that's exactly the reason why we wanted to go for a model of the TAC model, which is, kind of recapitulates the typical form of heart failure. Because even in FREF, you almost invariably have a component that is reduced or altered relaxation. And so we wanted to show that even in this model, by fixing the problem with the relaxation, we could improve also, overall cardiac function. And there is also a possibility of using then this peptide in combination with other drugs that may, beta blockers are used in HFrEF. And it is a little counterintuitive because in heart failure, you have already a weak heart. And if you block the receptors that trigger, that improve the contractility of the heart, you are definitely not doing any good to the ability of patients to move around, you don't improve their quality of life. And the reason we use beta blockers is because we keep the heart under control. We make it pump very kind of slowly and quietly so that it doesn't use up too much energy when this failing heart is already short of energy. But then using a specifically targeted intervention like the one I described, where one could, for example, think of elevating cyclic MP selectively around the Circa pump so that you pump calcium back into the sarcoplasmic reticulum very efficiently so that you have more calcium to be released. And at the same time, you promote relaxation. The combination of these two mechanisms could actually work potentially very well. And fix the cardiac performance without tiring the heart too much.
Thank you. Kenneth asks, how big is P33? And to that, I would add, how does one take P33? How is it delivered?
Yeah, so the P33 is 23 amino acids. It's a bit long. We would like, so the work that we are planning now, we are doing now is to try to shorten this peptide while maintaining the ability to do what it does, because shorter peptides, and also we are trying to change the overall charge of this peptide so that it can cross membranes more easily. The shorter the peptide, the higher the chance that it will get into the cell and get to the target where it needs to go. So we are planning work around this to try and improve the peptide. How the peptide will be taken by patients, we don't know yet. Again, this is work that we need to do. Being a peptide, and as I mentioned, the problem with membrane permeability, ideally we would like to deliver this peptide by mouth. But that's not always possible. It's not impossible, but it's not always possible with peptides. So more likely routes would be through injection in the muscle or intravenous. And of course, all of these are things that we will need to test experimentally as we go forward.
My last question is, what's the next step now?
So the next step is to find money to be able to carry out all these experiments. But in terms of experiments, the next step are exactly what I was mentioning now. So to test how well this P33 can cross the membrane, we have already started a study where we have shortened and modified the sequence of the peptide. We have already found a peptide that is much shorter and shows at least 10 times higher affinity for the troponin compared to P33. The next steps will be to test how well this peptide is stable in plasma, how well it crosses the membranes, and then we will go through all the evaluation of pharmacokinetics, pharmacodynamics. This is all preclinical work that we need to do to then move into maybe bigger animals, larger animals that better recapitulate the pathophysiology, the human pathophysiology. And hopefully, as we progress, we will then move into clinical trials if all goes well in the meantime.
Following up on that, John asks, how does the peptide cross the cell membrane?
So the peptide has a little, we tagged this peptide with a little myristoylated group, which makes it more hydrophobic. But we are also planning to change some of the amino acid to give more positive charge to the peptide. And then there are other quite well-established modalities of modifying the structure of the peptide to make it more membrane permeable.
Okay, we can give another few minutes for the last questions to come in. Could you potentially do this with, because you now expanded on one specific compartment within the cardiac myocytes, but you mentioned there is many things that are regulated by cyclic AMP, different proteins. And so do you have plans on doing that in other compartments as well?
Well, I think this is a very good candidate. I really believe this is a very strong candidate. So we'll need to try and focus on this one in terms of translation. But in terms of basic research, for sure, as you know, in the lab, we have several projects where we are looking at different locations and molecular interactions that are involved in regulation of different functions, for example, metabolism and so we are from the point of view of understanding basic mechanisms, definitely we are looking at other compartments.
Perfect. Thank you so much. Last thing I should add is also, Anthony, to answer your question about the online lecture. If you email the development office, they will send you a link. But yes, it will upload it, as I mentioned earlier, onto the YouTube channel. And the final thing I would like to mention is not to miss the next lecture of this webinar series, which is the 10th of March with Professor Sir Jim Mallison, a very brief history of yoga. And with that, I would like to thank you, everyone. Thanks for the questions. They were very interesting for me. I'm sure for Manuela too. Thank you, Manuela, for the wonderful lecture and the great insight about the science that is going on in Balliol and the frontiers that has been carried through. And with that, I will disconnect.
Thank you, Anna.
And thank you. Thank you very much.
Thank you.